Bienvenidos todos al seminario de hoy. Pues tuvimos el placer de que nos visitan Linda Amaral-Zettler y Erick Zettler en el Instituto Oceanográfico de Holanda, Royal Instituto Real de los Países Bajos para las ciencias marinas, ¿no? Entonces, bueno, pues están aquí de visita en Cistal y estamos aprovechando para que nos den dos pláticas, ¿no? Una es sobre la aspiraciones sobre el sargazo. Los dos tienen un background en biología y han estado trabajando temas de microbios, en plástico, sobre el sargazo. Y entonces, pues, todos los trabajos en los proyectos grandes de los que están. Gracias.
Buenos días, gracias por la oportunidad de hablar sobre nuestro trabajo. Lo siento que mi español no es bastante bien sobre in the United States for 13 years. I worked out as research leader of our in the Netherlands. And one of our large projects we've worked on for about 10 years now, which is plastic in the ocean. We've all seen plastic in the ocean. You know, it's a problem. I'm old enough that when I was younger, you know, it wasn't as much plastic. You know, I wrapped my sandwiches in paper and, you know, plastic's great stuff. It lets us do things like this, my mobile phone, my pants, my glasses. It's a great product, but it also has caused some problems in the environment. This room is here. And then let's say expanded polystyrene film that we picked up in the Sargassum, so north of Bermuda. And you can see it's just covered with all kinds of life. There's particles, there's algae, there's a here. So it's sort of like the biosphere is this layer of life on the outside of our planet, and it's the plasticiere is the layer of life that develops and uses plastic. That's the thing. Yeah. So we're going to get this problem. Yeah, it's great stuff. It started being developed heavily after World War II, 1950s or so, and this is a magazine in the United States lab in the 1950s, showing this great plastic stuff. You know, it's cheap, it's light, you can just use lots and throw it out in your dishes. It's great. But this is and this continued increase is the production of plastic, global plastic, in millions of tons per year. So recently, we see 400 kilos of plastic per year that we're producing globally. That's more than the weight of all human beings on the planet. And that's this problem, which the UN is calling a global. I was happy to say that on the CCL yesterday for the North, but it's really not too bad. I don't know if it's worse. Sometimes the beaches are pretty clean. That's great to see. So how do we study plastic in the ocean? Right, the easiest way, you know, it goes about 10 centimeters down to the water. So you pull it. Floating plastic. So once this goes in the ocean, you can see photographs of the bottom of the ocean. Some of these areas like the Mediterranean and things where it's just covered at the bottom. The really small pieces, we set that in situ. So it's basically and water from these stores makes sense of the so that means the really tiny piece of plastic that will go right through this pipe. And then of course the and that's what we don't do very much. It's very labor intensive to get the kind of stuff. Okay, so we from the real last 10 or 12 years. This is a very good area. 1,000 square meters of the ocean, so it's mostly small pieces. So what we call microplastic, basically liquefied molecules called microplastic, and plastic is the really small pieces that were just smaller it gets, the more difficult it is to do research. This is something that we work with finance. These are some of the areas that is a place where that's where we work basically all the way down to South America. Ecologists, we mostly work with the really small things. And these are electron micrographs. Various pieces of plastic we collected in the ocean. Each has 10 micrometers, each of these pictures. So we see a whole ecosystem on these plastic microorganisms system. These are diatoms, so these are then cyanobacteria, these are photosynthesizing algae, so those like producers taking the sun, producing organic matter. And we have this is a ciliate called a septorium. So they attach to the surface of plastic almost like a tiny little lemon and these little tentacles, and they grab other protists and eat them. And if you see them on the surface of these, they're actually covered with even smaller microbes. These are bacteria that are somehow associated with this. So we have phototrophs, we have stigmatids, and then these are bacteria we call pinworms. We don't know what they are yet, but if you notice at the surface of the plastic, it looks like they are embedding themselves in little holes in the plastic. So the question is, are they actually digesting and metabolizing that plastic somehow, or are they just physically eroding it away? Of course, that's an important question to determine the fate of plastic. If we put all this plastic in the ocean, we know it's going to last a long time, hundreds, maybe thousands of years. We don't know how long, but the microbes are going to be for part of everything about it. So finally here, about stuff on plastic in the ocean, there's some things floating around the ocean forever. You know, wood, sargassum, all kinds of things floating pumice from volcanic eruptions. So plastic is just another surface well. The problem with plastic is it lasts longer than these other substrates. It could last, more than 10 years out there. Because of the serial number of weight. One of the basically species, this is native sargassum, these colored sargassum. Well, now it's trying to move on to pieces of plastic. So the sargassum has a certain distribution in the Atlantic. A piece of plastic you can see we drift much further and introduce species that are native to different areas of the ocean. Most of the certain potentially dangerous disease causing microbes attached to plastic. This is bigger this is species, a genus of bacteria. The one you might be familiar with is a bigger perteneciente, which causes a lot of shellfish poisoning when you eat shellfish. Bigger cholera causes disease cholera. Actually, So these are the reasons we kind of want to know more about the microcommunity. Are they plastic? Are they potentially transporting dangerous organisms around the ocean? All right. So this, over the years, the different sort of ecological questions. Microbes are really small. Theoretically, they can be dispersed anywhere. So would you find the same microbes on plastic no matter where in the world you sample? These are two crucial facts we did in the Pacific. Linda's so you saw from those pictures, the bacteria is a little so we do extract the DNA, and she analyzes the DNA to look at the base pairs and the genes, and from that she can identify at least the group of bacteria they belong to, and look at diversity and maybe who's on that plastic. So if we do that with this data set, now each symbol is a single sample looking at the microbial community setting. You compare two samples using multivariate statistics. These are, what are these? Are these PCA or what are these ones? Remember?
PCA.
Yeah, these are okay. Well, PCA. So yeah, so this is a multivariate. Each of these is a sample. The closer two dots are together, that means the microbial community is more similar. The further apart those two symbols are, the microbial community, both diversity and abundance, was quite different. And what we see is the blue ones, the Pacific, separate out. They're not overlapping. It's not just one big cloud. There's some overlap. There's some red ones down here, but basically there is a difference. Geography matters. Where you are in the ocean changes what the microbial community is. Why does that matter? Well, then if we do an experiment in one part of the ocean and find out, okay, the microbes break down the plastic at this rate, you can't necessarily say the same thing will happen in the Pacific or in the Gulf of Mexico. So the geography matters. This slide is looking at it instead of the different polymers. Once we extract the DNA, we use Raman spectroscopy to look at to determine what kind of plastic it is. We take the same data set and plot it based on different kinds of plastic, polyethylene, polypropylene, polystyrene, and PET, those bottles. You see that there really is no pattern. They kind of just all belong together. So the plastic type doesn't matter as much as the location. Thanks. Another simple experiment we did early on. You just take different kinds of plastic. These as our control, polyethylene, polypropylene, polystyrene, just common consumer products. So a milk jug, coffee cups, cold water cups, and we put them in a mesh bag so that crabs and things can't eat them and break them up. And we just put them in the dock, in the water at different places. You can do it right out here. And over time, we sample the different kinds of plastic and look at the microbial community. So if we do that, if you look at electron microscopy after 1 week, it's just coated with these little oval cells, which are those diatoms, those photosynthetic diatoms. After 2 weeks, you still have diatoms, but you have other things coming in. And after 4 weeks, again, you still have diatoms, but now it's really starting to grow into a three-dimensional layer of microbes coming off that plastic. Again, if you look at the molecular data, look at the DNA, you can see early on, triplicate samples, lots of diatoms, that makes sense. Still diatoms are fewer and still diatoms are fewer. What you do see, there's some changes. This one here is a cyanobacteria. It's fairly abundant here. It decreases, as opposed to this one. It's pretty much constant the whole way. So there is an evolution in these communities over time. Next slide, please. And if you plot that again, using principal coordinates analysis, PCOA, again, if symbols are close together, the community is similar. And now we're plotting different kinds of the different polymers. So week 1, the replicate polymers sort of cluster together, more or less the same. So here, the type of plastic does make a difference. But as you go to week 2, week 3 and 4, and week 8, you start to lose those patterns. What we think is early on, when it's bare plastic, the microbes are actually experiencing the surface of plastic. So the chemistry and the physical properties of that surface make a difference to who settles there. But over time, you have just a layer of of microbes. And so after that, all the plastics look the same. So we think that the communities start out different and then converge at the end. Excellent. So more recently, what we started to do is we want to know, okay, with these newer, you've probably seen in stores and on advertisements, biodegradable plastics, right? They're okay because they biodegrade. They turn into carbon dioxide and water. So this is a respirometer system we have in the Netherlands. Basically, we can take bottles, put seawater with different types of these new biodegradable polymers like polyhydroxide, PHA, PHB, these different things you see on bags and soda bottles, and put them at different temperatures and actually measure. What these tubes do is they take all the gas that's coming off the top through these CO2 sensor and a methane sensor, so we actually monitor the CO2 production. What that tells us is we have a blank, which is just seawater. So if there's more CO2 coming off the one in the plastic, that means that the carbon in that plastic is actually being metabolized and converted to CO2, so we can actually measure the breakdown of plastic. And actually happens quite fast. Next one. This is, sea temperature makes a difference, right? 10 degrees centigrade, 20 degrees centigrade, same seawater, looking at different polymers. So this is cellulose. The blue is cellulose. We use that as basically just, paper. We use that as our control. At 20 degrees C, that basically is mostly broken down after not very long, 300 hours. So it's really quick. Much slower at 10 degrees centigrade makes sense. But even some of these other polymers here, that's the blank, that's going to be the blank of just seawater, not very much CO2. And then this one is our PHA, our biodegradable plastic. And that is also going down not as fast as cellulose, but still significant breakdown. What we found that at 20 degrees or so, approximately 60 to 70% of that PHA will be converted to carbon dioxide in just 3 to 4 months. So that's actually pretty quick. But what we're interested in now is what happens at different temperatures. These things sink. Most of the new polymers are denser than seawater, and they sink. So they're not going to stay at the surface. They're going to go to the bottom. And you guys should know, what's the temperature at the bottom of the ocean? Someone help me out here. Come on. Cold. Yeah, exactly. Like 5 degrees centigrade or less. So what we want to know is, okay, this is all great at warm temperatures, but what happens when that stuff falls to the bottom of the deep ocean? So that's the kind of work we're trying to do now. Thanks. And just a quick, again, electron micrograph. I love electron microscopy. It's like magic. This is the surface. Here's 10 microns. This is the surface of that PHA. After about 4 weeks, it's just loaded. All these things that you see here are bacteria, filaments, and things. So this thing is just loaded, and you can see it's being eroded away. Little pits and holes. This is the surface of plastic, and all this is biofilm. So these things are really love munching on that stuff. And I think I'll probably skip through this part. This is probably more than we need to know. Well, basically, when we do electron microscopy, what we do so far, this is a diatom shell. These are the bacteria. But we have no idea what kind of bacteria those are. So what we're doing now is we're using the molecular data. We know the DNA. We extract the DNA, and we make fluorescent probes. So things, little colored probes that attach to different kinds of DNA. And then we can look at the fluorescent microscope, and now we see that, okay, these all look the same here, but each color here is actually a different probe telling us these are different kinds of bacteria around the diatom, and we can see that these yellow ones are more common. So I think I will quit that. This is too much. This is what the microscope. This is the confocal microscope. We managed to get it because the key of it here is this is a spectral sensor, so it actually has 34 different photomultipliers, so you get a full spectrum of each sample. Really isn't too much. So basically, what you do is you train it so that, okay, there's one kind of cell, there's a second, train the computer, there's a second kind of cell, there's a third kind of cell, then you tell it to put them all together, and you can overlay them and see the 3 different kinds of bacteria that we're looking at. I think I'll quit there because that's probably as much as I want to say about plastic. And just thank you. I think maybe better to wait till the end for questions and let Linda speak. But thanks for your attention. I look forward to anyone that wants to chat later or ask questions. Happy to talk to you about it.
Sargassum ecology and it's partially my answer. Okay, so as Erick mentioned, we spent most of our careers in the Marine Biological Laboratory in the Woods Hole Geographic Institution in the village of Woods Hole. And in 2017, we moved our group all the way to Texel in North Holland, rather. And now this is my group. As I said, we focus on sargassum, but also marine aspects of marine plastic biodegradation and also nanoplastics. And I also maintain still a technical colleague at the NBL, and so I routinely go back and forth across the Atlantic, but our major focus now is in the Netherlands. Good. Okay, so we're going to shift now then from what I call plastic tides, and sadly, all these little black pellets you see are beads. This is the result of a switch overflow, likely from the UK, where these little plastic beads that are used in sewage treatment are released. And these are all beads that are littering one of the beaches in the Netherlands. So we're going to shift from plastic tides to what we call golden tides. And I know that here in Mexico, you're very familiar with these sargassum inundations. This particular image is from Curacao. So sargassum is, in fact, a nickname of the golden floating rainforest. Of course, in the open ocean, it's really a haven of life. And this is an image from Barnes that shows it's not only sargassum, right? It's this community, this interesting community that's attached to sargassum. And of course, within these invertebrates that inhabit the sargassum, we see this incredible microbiome as well. So a very, very rich and abundant life that occurs in the sargassum. And I love this artist rendition of some of the endemic taxa. Erick talked about this in his previous slide in terms of plastic ghost nets now adopting some of these endemic taxa. But again, a very rich haven, if you will, a nursery for many, in fact, endangered species themselves. And so there was a real movement back in 2014 to come together and protect the Sargassum Sea, right? The Sargassum Sea is the namesake. Sargassum is the namesake of the Sargassum Sea, and several nations signed on to this agreement. But of course, here we know that there is a dark side, right? There's a dark side to Sargassum, and these are some of the headlines from several years ago, including one from Mexico, calling on the Navy to combat this piece, if you will, of an entity. And so that's about 2010 and prior, we started becoming very interested in sargassum, and in particular, the Sea Education colleagues there started measuring sargassum in different parts of the Atlantic and started noticing something very interesting, and that is traditionally there were two morphotypes that dominated the parts of the Atlantic, natans and fluotans, but there was a different morphotype that started appearing back in 2011 and even beyond that. And so there was a question as to whether these inundations that started happening back in 2011 were somehow related to climate change or perhaps even a natural range expansion. And oops, I thought I had another image here, but so much so that they started entering into countries such as Brazil that had never seen sargassum before. Going wrong way. Oh, no. So again, Brazil had never reported sargassum. So for the first time, it was starting to see this seaweed. And again, the reason this is relevant, of course, in addition to the damage it is doing, is the potential for introducing exotic species to Brazil, which of course is very protective of its biodiversity. So one of the things we did early on was to look at, along a transect of sargassum, to better understand how different genetically some of these morphotypes were. So each one of the morphotypes that I mentioned in my previous slide, they have very distinct morphological features, but we wanted to know if genetically they were also distinct. So we collected sargassum along this cruise track that I showed you here. And we were able to sequence the mitochondrial genomes of the sargassum. And we actually went to the herbarium at Yale University to look at the different morphotypes, which are the voucher specimens are collected there and stored there. So we were able to directly compare the morphology and the genetics. And this is actually a phylogenetic tree that shows effectively the genealogy of the sargassum. And you can see that these are the two sargassum natan forms, and this is glutans. This is a tree based on all the sequences in the mitochondrial genome. And what I can tell you, we published this back in 2017, is that they are very, very closely related, amazingly closely related. And yet, there are stable polymorphisms that are unique to each one of these morphotypes. So they, in fact, are genetically distinct, albeit extremely closely related. So when I arrived at the NIOZ back in 2017, I was granted a cruise to further explore the diversity of sargassum, and in particular, target this area here known as the Great Atlantic Sargassum Belt. And so this was a cruise that we did from the Azores, right here along the Mid-Atlantic Ridge, all the way to Guadeloupe, sampling along the way and all these white dots that you see. And this here is our vessel, the Pelagia, which is the Dutch national oceanographic vessel. And the image I showed you on the cover was, in fact, taken right around here. So this was not a coastal image. This was actually in the middle of this Great Atlantic Sargassum Belt. And during this cruise, it was really amazing because these are satellite images that were produced by Trim and Cruise Group in Florida. And they helped us track these patches, these huge patches of sargassum during our cruise to be able to better study the ecology and distribution of sargassum. So one of the overarching questions we had at the time is, where do polypelagic sargassum originate? Now, you're familiar with the Sargasso Sea as being the original home of sargassum, but in fact, these huge accumulations here, and now it's called the Great Atlantic Sargassum Belt, was really the target of this cruise. We wanted to better understand, okay, is there variation in the types of sargassum that we see? Are there any clues as to what's going on in this part of the ocean? And you can imagine, and this is a very remote part of the ocean. I think we may have seen 2 other vessels during the entire cruise. So it's not a well-frequented part of the Atlantic. And it's hard to get to. It takes a long time to get to this area to study it. So, like with plastic, we have ways of studying sargassum in the open ocean. In some cases, we collect via zodiac to be able to preserve the health and morphology as much as possible. In other times, we use dip nets, or we use the manotrawl, which I showed you earlier, which is what we also collect plastic with. And part of the cruise was dedicated to microbial ecology, genetics, and ecophysiology. We also were looking for remnants of sargassum in the bottom of the ocean. So we took what were called sediment cores. This is a box core that we took to be able to see if we could identify sargassum at the depths of the ocean. And in fact, we even found sargassum down to 4,000 meters below the ocean surface. So indeed, sargassum is present even in the very depths once it dies. And we were also very interested in chemistry. I think people are aware that sargassum has a propensity for accumulating heavy metals. And so there was a group on board, a student actually from Guadeloupe, who was looking at heavy metal concentrations. And one of the interesting findings that she published as part of her thesis, this was Oceana Cipolloni, she found that, in fact, these different genotypes, this is Sargassum daytans 1, Sargassum daytans 8, and fluitans, did segregate out based on the abundance of certain kinds of metals. So in this case, SN8 had more arsenic associated with it, whereas SF3, Sargassum fluitans 3, had more iron associated with it, for example. In addition to chemistry and biology, we also looked at the physics of sargassum. And let's see if I can go. Okay. So this is a little movie that demonstrates these what are called Stokes drifters, which are drifters that are designed to mimic the movement of sargassum. And one of the interesting observations from this study that was conducted in collaboration with the Dutch colleague Erick Zettler was that these new drifters that we put out were better representing the movement of sargassum in this part of the ocean. So we are planning to do more of this kind of work to better predict the movement of sargassum itself. And we also were very interested in these attached macrofauna, and we worked collaboratively with Veronica Velasquez, who is one of your own here at UNAM. And what we did is, this is another type of multivariate analysis looking at differences in the attached communities, the macrofaunal communities between the three different kinds, the three different types of sargassum, and found, in fact, they were also distinct. So not only is the sargassum genetically distinct, but you can see differences between the communities attached to them. So current work involves my student, Tom Terling, who joined us. This is the first cruise that he did in 2019. And on board, we were able to do growth experiments with different manipulations of nutrients and dust. We also looked again at the microbiome of sargassum and also at genotyping the sargassum itself. And we came up with a very simple way of differentiating between the three different genotypes. And we're able to even do that on shipboard, as you can see, the two of us working here, extracting DNA and genotyping on board our vessel. One of the things that Tom discovered in looking at the microbiome is, in fact, there is a lot of shared. So this is the Venn diagram showing the different types of bacteria associated with the different sargassum. And we see again that there is a lot of shared taxa, bacterial species, but there are also very unique ones indeed as well. And there are some differences in the diversity across the different genotypes. But we're taking that one step further because the mitochondrial genome is just not very adequate for differentiating between potentially populations of sargassum. So we've been sequencing using traditional, well, now traditional short read sequencing methods like is available through Illumina, but also PacBio sequencing to get the entire genome. And so this is a work in progress, but we do have a draft genome of Sargassum fluitans as well as Sargassum fluitans as well as Sargassum daytans 1 and 8, which are not quite as complete as Sargassum fluitans 3. So back in last year, we were awarded a grant from a coordinated effort between the ANR, which is the French Science Foundation, and the Dutch Science Foundation, and FACETI, which is the Pernambuco division of the Brazilian Science Foundation, to form a consortium called the Wheels of Change. And this particular consortium is looking at characterizing and modeling the sources and sinks of the Great Atlantic Belt. And you can see here, this is Rita van Tussenbroek from UNAM, who is joining us from her own collaborative sources to be able to look at different aspects of sargassum, including growth and utilization, biophysical modeling, in particular looking at the growth aspects of modeling sargassum, and also biomarkers and geomarkers. And my group is overseeing, and I'm the consortium leader. We're overseeing all this work. And at the heart of the project is really a cruise back to the Great Atlantic Sargassum Belt, where we're going to be repeating some of our original experiments with a bit more knowledge that we gained from doing coastal experiments. Rita is really organizing a lot of the coastal growth experiments that are going to be done in Guadeloupe, also in Brazil, and potentially other parts of the Atlantic as well. So our next cruise is planned. So Eric, in fact, has already sailed from Vigo, Spain, to the Bahamas, testing some equipment that we're going to be using on board our cruise, including a trap that will not only allow us to visualize sargassum sort of in the horizontal plane, but in the vertical plane. So we're going to start looking at how sargassum sinks, as well as how it disperses. And this coming June, July, we have our big cruise that will go in the opposite direction. So the original cruise that we did back in 2019 went from the Azores to Guadeloupe. This time we're going to go from Guadeloupe to Cape and again, repeat some of the growth experiments that we did earlier, looking also not only at addition of nutrients, but also upwelling water and a few other variations. In addition, we're going to be looking back in time. We're going to try to look to see if we can see how far back sargassum blooms have occurred in this area, if there's any precedence for sargassum being abundant in this area in the past. But another thing that the sedimentary record can tell us is what the environmental conditions, how they've changed in areas where sargassum is now blooming, for example. We will also be deploying more sophisticated drifters that even mimic the movement of sargassum even further as part of the cruise plan. In 2025, we have a special cruise. It's a short cruise. It's a 10-day cruise that is associated with a course. We're going to teach a master's course for 4 weeks, about 4 weeks, 2 weeks, well, 10 days on our vessel, a vessel. We don't know which one yet. But then also a short component. And this particular master's course is going to be open to at least 4 students, hopefully from the Caribbean and in sargassum impacted areas. So be aware and tell if you have students that might be interested in this. It is the title is Seaweeds on Shore and at Sea, but it's really dedicated to sargassum and sargassum research, both looking at the ecology but also uses of sargassum. It's going to conclude with an expert workshop where we'll engage stakeholders in different aspects of the issues surrounding sargassum. And as with our first cruise in 2019, we have blogs that we typically do. If you want to learn more about the work that we did back then, you can still read our blogs. They're available on the NIOZ website, and we're hoping to have similar blogs available as part of the upcoming cruise that we'll be doing in June and July of this year. So I will end there. Thank you for your attention and this opportunity to share our research.
Thank you for the presentations. Regarding the drifters, are they attached to the sargassum patches, or they are free?
Yeah. So originally we were thinking of producing tags that would actually be physically attached to the sargassum. We've kind of stepped back a little bit from that idea by a design, or I guess adopting a design that has a sort of a net material that kind of attaches, gets stuck. It gets stuck, and then it would presumably go along with the sargassum. I don't know, Eric, if you want to talk.
That's enough. This data that was already published was just the drifters.
Yeah.
And then exactly as you say, we realized they don't always stay with the sargassum. They're more similar than a drone drifter than typical drifters, but so now we're trying to work towards what you mentioned and just try to figure out, and we have some colleagues that have done a couple experiments working on this. It's not perfect, but it's better than what we did last time. The other exciting thing about these new sensors or new drifters is they have some sensors. They're going to have light, temperature, and in a couple of cases, not too many because it's very expensive, an actual TCO2 to measure the partial pressure of CO2 in the sensor under the Sargassum mat. So, little step, not perfect, but trying to get a little more realistic each time. Yeah, that's a challenge. Right. Sure. Yes.
So, for the bacteria attached to the microplastics,
Yes.
do you think they attach because they use something in the microplastics for food or for something, or they just attach to the bacteria to move around?
That's a great question, and yes, there's certainly. It's the answer is both. There's a lot of bacteria that just attach to any surface. You know, cement, a stone, a piece of wood, plastic, generalists that they're just looking for a surface to attach to. What we find is that's most of the bacteria, but there are certain groups, a subset of that seem to be more abundant on plastic than on other surfaces. So they're somehow they prefer the plastic a little bit more over other substrates. With conventional plastics, petroleum-based plastic, polyethylene, polypropylene, the biodegradation is so slow, we don't think that's we think it's more of the physical properties. But with these new biodegradable plastics, there's definitely, some of them are made from, PHA is a naturally produced bacterial product. So the bacteria that produce PHA is a carbon storage product. And so we know they can break down PHA, so they will definitely be more abundant. So it's a little bit of both, but yeah.
Yeah, and maybe I can add to that. So the idea is that if you have a virgin plastic, something that's newly produced that is perhaps not weathered, and is subjected to UV light, it will produce effectively leachate products that in fact are very digestible. These are short chain carbon molecules that are basically being released and are effectively edible by bacteria and utilized by bacteria. The other type of potential utilizable material are additives. So, right, because if you think about plastic, we think of pure plastic. There's no pure plastic out there. There are some if it's a pellet, but that's a rare finding now, especially. So it's usually these, there are plasticizers, there's all kinds of additives added, and in that case, you could easily have bacteria that are capable of breaking down those materials in the absence of being able to actually break down the carbon chains that are very, very, very difficult chains to break.
I feel like doing a nice experiment. Just take one of these bottles and a little bit of water and put it on a sunny windowsill for a day or two, then open it up and smell it. You smell all those organics that have come off that plastic from the additives. Yeah, exactly.
All the stuff.
Yeah.
It's really, really interesting. I work with ecotoxicology.
Oh, okay.
Environmental chemistry, and I never thought of bacteria jumping and adding to that. So it's really interesting stuff what you are doing. And also, in my field, there's also a debate that, the plastics are also like a substrate for pollutants to move around. So now I'm thinking, well, what if the bacteria also help to move pollutants here and there?
Yes.
So it's really interesting. Another thing that I was thinking, once I went to a cenote, the sinkholes, and I had to put a trap there for any other stuff not related with these topics. And I remember I looked up and the cenote was covered with soda caps all over the cave. So now I'm thinking, what if you take out the light and the salinity in that process of degrading or not? The change. Yeah, that would be really interesting. Completely different environment.
Yeah.
No sunlight.
Yeah.
No salt.
Same at the bottom of the ocean, no sunlight, cold, high pressure.
High pressure.
Yeah.
So very, very interesting.
Have you comment on that?
Okay. Thank you for the presentation. I'm looking at biology. I want one question. Why the chloroplast decreased for a longer time?
Over time, what are the diatoms we think are just, they're very early colonizers. So when a new substrate, new surface comes in the water, they're the first ones to colonize. But then over time, other things start coming in and outcompete them. So they're still there, but they're not the only ones. And so that's why in a relative sense, there's fewer chloroplasts over time, because other things are coming in and taking their place.
Okay. And other bacteria increased.
Yes, exactly. Yeah. Bacteria increase, the diatoms with the chloroplasts go down.
Okay.
In a relative sense, yeah.
There are also potential pathogens and bacteria that can splice the diatoms as well. So there's all kinds of interactions going on that we're trying to study, but again, it's difficult because the biofilm is very complicated. And when you extract the DNA, you sort of destroy the structure. So that's why we're using microscopy to understand more, okay, well, this organism is next to this one, what's going on? This one is inside of that one, it got eaten. And these kinds of questions are ones that we're trying to better understand.
With that climate change and changes in temperature and things like that, link it all with the biodegradation. Do you think biodegradable plastics are actually, because there's a big argument at the moment, isn't there? Replacing plastic with biodegradable isn't an ideal solution either.
No.
But from what you're showing, it looks like biodegradable plastics are helpful. What do you think with climate change and everything else? Are actually beneficial, or just switching plastic for a completely different product, like plastic to be reused?
Yeah, well, we always say biodegradable plastics are not a solution. It's a part of the solution because even in the best case scenario, you don't get 100% biodegradation. So it's always going to be, what we're saying is it may be one stopgap method to decrease the amount accumulating in the ocean. I mean, the best way to reduce plastic in the ocean is just don't put it in there, right? So that's really where we have to make the greatest effort. Certainly in coastal areas, as temperatures increase, biodegradation, our needs equation, chemicals reactions go faster. So yeah, it's going to speed up. Whether that's a good thing or not, I guess we're getting rid of plastic. It's good, but
Using more CO2 at the same time.
what's that?
Using more CO2 at the same time.
Exactly. Yeah. Well, I guess the other advantage, what they claim with biodegradable plastic, if it comes from a biological feedstock, the net CO2, it's not taking, buried CO2 out and releasing the staying CO2 that was already in the atmosphere, re-putting it in the atmosphere. It's not great, but it's at least it's a net, theoretically a net zero.
And I mean, I think the thing to remember too is that there are applications where plastic does escape into the environment. And those situations will always exist. But right now, it's really recalcitrant plastic that's being released. So as opposed to using those kinds of plastics, replacing them with more environmentally friendly plastics is one idea. I think the other issue is waste management and the fact that as well as we can manage waste in certain parts of the world, that's not the case in many places. And so biodegradable plastics does lend opportunity for controlling the amount of plastic that ultimately accidentally gets released into the environment. So I think those are two issues to really be thinking about. I think the challenge is making sure that these materials really are biodegradable and preventing greenwashing.
That's why we're doing this.
How is it like how biodegradable? Actually, they've stuck on lots of things which actually, but the opposite of biodegradable, which is taking longer and things like it's biodegradable and it'll be right. But
Exactly.
actually, it's not 30 days, it's 30 years, but it would be great.
Yes, exactly.
Yeah, and that's where, the world has to come together, and it really isn't, because it's fine if you, deregulate a certain kind of plastic in one country or even one continent, but the ocean is everybody's, right? And so it doesn't work that way. So this is a challenge before us.
like the question with the, you said something about samples from 4,000 meters. Yeah, used to concept. My master's thesis was on chemosynthetic requirements. But you have any samples from any area that are near anything?
No, we don't have any samples like that. We really, I mean, as you know, and if you worked on that, it's really hard to work down there. And so we're just starting to scratch the surface of.
It'd be interesting to take advantage of some of the dives they do and see if they can get samples of people's there and if it's a great thing.
Linda has a proposal in now to try to start doing a little bit of deep sea
Or in the deep
work.
sea, yeah,
Because it's just,
it's a huge question mark. Obviously, it's very difficult to get down there and study, so, but yes, we are very keen.
and we know a lot of plastics ending up there, so it's a great question, yeah.
Yeah, especially with the Sargassum, because we found that with the, we were studying before the mussel, reach out using mussels. And from the saline lakes and also from to see whether they had a relationship between the primary production on the surface and what reached the bottom. So if you're looking at this plastic tied up with that actually.
Yeah.
Yeah. So that's very interesting.
Yeah, thank you.
So I was just finding any similarities between the biomes in Sargassum and plastics.
Yeah, so there are certain bacteria that like to colonize substrates, and sometimes you do see similarities there. But in the case of Sargassum, there does seem to be, well, it's thought that Sargassum controls the microbial community by producing chemicals that regulate who can settle where and when. Yeah, that's what I can tell you. Although we have found, for example, embryos like plastic and they also like Sargassum. And so that's been an interesting observation, particularly in the Sargassum, in the case of Sargassum, as the Sargassum starts to decay and dry out and strand, Sargassum, the embryos would really increase in abundance. The phenomenon that we sort of did the same initial cruise, Sargassum cruise we did, there was a simulated stranding experiment that one of our Brazilian students did in Mendoza. And basically, you see this over 24 hours, this tremendous shift from the much more diverse bacterial community to one that's dominated by embryo. So it's a spike. And we've seen similar things with, well, we've seen some naturally, quote unquote, occurring plastic with increased numbers of. And part of it is growth rate. Embryos are really fast colonizers and also grow really fast. So depending on when you sample whatever material you have, it might be reflective of this. So, but it's complicated. It's a complicated question.
So if you have the diatoms, use the DNA test, look at the DNA of the diatom.
Yeah, it depends. Or the bacteria, eukaryotes and the bacteria in the archaea, you can really design your experiment. Now we're also just sequencing everything. So we're also editing. So everything that's there, trying to reconstruct the individual gene. Of course, with the eukaryotes, it's much harder, but it is also possible.
More questions? Okay.
Thanks, everyone.
Got it.